Okay, I would like to start with thanking the organizers for getting me here, and thanks Madhu for the kind intro. So I'm Rishi. I am from the Indian Institute of Science. That's the main tower building of our institute. Right. So let's start there, and I think the topic has nothing to do with either sensory side of it or motor side of it. So that's the good thing to start with, as it were. Right. So we'll start with it. So I say functional maps within a single neuron. So it is imperative that I define what a map is. So a map, so you have a certain set of inputs in a set that is shown over here, and there is a set of outputs that is represented by this yellow color wiggly surface sitting over here. And if you have a topographic organization of things mapping on from this set to this set over here, in a certain, so adjacent locations from this set basically map onto adjacent locations along to this set over here. So it's like a map which is representing real space, or in neuronal jargon you could call it as either say an auditory system where you have different locations along the neuronal tissue or responsive to different frequencies of auditory stimulus. So that is an auditory map that is sitting inside the auditory system, or you could have a visual map over here. So either you have various different maps in the visual system. You have the retinotopy, where you have adjacent locations in the neuronal topograph, neurons located adjacent to each other, or mapped onto locations which are adjacent to each other in the visual space. And you have here orientation maps, where if you traverse along a certain direction, as long as you don't get into these singularities over here, so you will see that there is a smooth gradation of various different orientations. These different colors are representative of different orientations, and as you traverse along this line over here, you see that the orientation selectivity of neurons sitting at this space is going to be slowly varying, like this thing sitting over here, where you have different neurons responding to different frequencies, and this end of it there is a low frequency neurons responding to lower frequencies, and this end of it you have neurons responding to higher frequencies. So this is what we call as a map in neuronal jargon, and we all know that sensory processing occurs in well-organized neuronal maps. What we perhaps do not know is that that is the case even within a single neuron. So this is an example of a neuron map that exists within a neuron. So here what is shown is that you patch on, you make wholesale voltage clamp, wholesale current clamp recordings at different locations along the neuronal tree, and present various different frequencies to this neuron sitting over here. You measure the impedance, which is the voltage response that you get divided by the current that you inject over there, and the impedance amplitude profile would look something like this, where you have a distinct resonance where you have the neuron specifically responding maximally to this specific frequency, which for these neurons, the CA1 hippocampal pyramidal neurons, that range is usually in the theta frequency range, which is around 3 to 10 hertz range basically. Right? So you have these neurons responding to different frequencies, but it's not like the resonance frequency at the soma is the same as the resonance frequency at the dendrites. If you move along this topograph, the somatoapical trunk of this neuron sitting over here, you will see that adjacent locations respond to adjacent frequencies, and there is an increase in the response frequency. So the somatic membrane is responding maximally to around 5 hertz, and at the distal end at around 300 microns, you have that responding to around 10 hertz over here. Right? So you have a map of different resonance frequencies along this neuronal topograph, and you have adjacent locations responding to adjacent frequencies, as it were, pretty similar to what you see over here in this case. Right? So it is a map in some senses. Right? We also know that in the visual system, it's not like there is one map sitting over there. You don't have just the orientation selective map. It is as though the same neurons sitting in the V1, the primary visual cortex, also respond to different directions, and there is a direction selectivity map also, which is sitting along with this orientation selective map on the same neuronal structure. Right? So this is orientation selectivity, this is direction selectivity. And you also have the ocular dominance column, the retinotopy, which is being mapped onto the visual field being mapped onto the retinal structure and beyond, that is also sitting on the same map over here, right? So we know that there are multiple maps within the same brain region involving the same neurons that sit over within that structure that we see. So is the case with simple single neurons as well. It is not as though in this hippocampal pyramidal neuron somatodendritic axis, the resonance frequency map is not just the single map that is sitting over there, right? So you also have, so if you measure the amplitude of action potentials at different locations, so as opposed to the Kahlian view where you have the distal dendrites don't get the action potential that is generated at the cell body. We now know that action potentials do back propagate into the dendrites apart from going the usual way along the axon. You also have the action potentials propagating back into the dendrites, and their amplitude reduces with distance, not because of a passive attenuation that Rahl proposed, but because of a gradient in A-type potassium channels that increases with distance along the somatodendritic axis. So you have, again, if you look at the amplitude of the action potential and map it along this topograph, you could envisage this as a case for a map, right? You also have this neuron which seems to know cable theory in some senses. So the neuron seems to know cable theory that, okay, if I get an input over here at this location, then it is going to be attenuated heavily as it propagates through this entire cable structure before it reaches the soma. And if I need the same amplitude of the EPSP at the cell body, irrespective of where the input is coming in, then I should increase the local EPSP amplitude such that the propagation will compensate for this attenuation that is caused by cable theory. And you have the local EPSP amplitude. So if you measure the local EPSP amplitude at the dendritic location, that is going to be larger than what you get at a proximal location in the same neuron. But at the cell body, because of the attenuation that is caused by cable theory and other active processes sitting over there, you see that the amplitude at the soma is kind of maintained. What Michael Hauser calls this is as dendritic democracy, where irrespective of the location of the synapses, all of them get equal representations. So it's as though the distal synapses, as he says, shout louder for them to be heard at the cell body, so that you have equal amplitude. Right? So these could also be construed as maps on the same neuronal topograph sitting over there. So you can see that multiple maps are present along the same neuronal structure. Right? So these are some of the examples of the maps. So that's the backpropagating action potential amplitude, the EPSP amplitude, and you have various different resonance frequency mechanisms. So the H resonance frequency, the one that I showed, increases with distance from the cell body. M resonance, which expresses at a slightly more depolarized voltage, and it's because of what's known as the M-type potassium current, that reduces with distance from the cell body basically. And this is input resistance, which again is mediated by the presence of certain ion channels. Right, so all these maps within the, right, all these maps within the neuron are mediated by one or the other ion channel. Right, so there are distinct gradients of different ion channels that are expressed within the dendrites of these neurons. So say, for instance, if you look at the CA1 pyramidal cell, you see that there is an increase in the H-channel density, even though it is shown to be linear over here. We believe that it is not going to be linear because you saw in the previous case that this is more like exponential, right? So if you need this kind of thing, theoretically, you would need something which is not linear, basically. Right? So it increases with distance, all right, but we don't know the exact profile of this function that would characterize the density of H channels on these CA1 pyramidal dendrites. So you have the resonance frequency map mediated by these channels, which are expressed in the dendrites. And the backpropagating action potential map in the CA1 pyramidal cells is mediated by an increase in the expression of the A-type potassium current sitting in the dendrites. So because this transient potassium current is higher, the sodium-potassium balance is tilted in favor of potassium, and hence you have a lower action potential amplitude at these locations. Right? So these maps within neurons are mediated by the presence of ion channel gradients that sit on the somatodendritic topograph. So the question then is, as somebody eloquently put it, why, right? So why do you have these maps sitting over here? What is the physiological relevance? I mean, the role of physiologists, which used to be the dominant topic a while ago, but now we have to define physiology as the concept of putting molecules together, and it has taken a new avatar. People call it systems biology, and you can refer to that basically. Anyway, so what is the physiological relevance of these maps sitting over in these dendrites? So each and every map has its own function. Say for instance, you have the AMP receptor gradient is there to counter cable theory, as it were. It is as though the distal dendrites know that there is a cable equation sitting over here, and I have to amplify this much for me to be able to get to the soma at the same amplitude. And the resonance frequency map could be construed as a mechanism by which where you have different parts of the neuron responding to different frequencies. It is as though each and every compartment of this neuron is tuned to a separate optimal frequency, right? So each and every of them have different theory, I mean different function, all right. But the question is there an overarching theory as to why all of these are present in the same gradients within a single neuron? So all of these are not present in different neurons. We are studying them differently, but all of them are sitting in the same neuron doing something as a collective ensemble. The question is, what is that it is doing, right? So we'll start with the more well-established sensory map literature and see if we can get some clues from there as to why this should be happening within neurons. So in the sensory map literature, there are two prominent hypotheses. When I say prominent, it basically means that there are more, and I'm just emphasizing two of them. So one of them is the wiring rate optimization, right? So which was proposed by Chaklowski and others, a series of papers which came in the early 2000s. So this is an example of why you would need a retinotopic organization in the visual field. So the argument is that if you want to extract local features from a specific location, you need interactions between neurons. Let's say that each of this square is a receptive field for a single neuron that is sitting in the visual cortex. And if you want interactions between these different neurons sitting over there for you to be able to extract a local feature, then it is better to have these located at close by locations in the primary visual cortex rather than having a non-retinotopic organizations where each of them is dispersed all over the place. So if you want to extract local features, which is one of the prominent ways by which the visual cortex functions, I mean, you go from simple center surround receptive fields to orientation selective receptive fields, and you have like face recognition schemes which are coming down in the lane at IT and other places, right? So if you have a non-retinotopic organization, and if you do want to extract local features, then you will have to have longer axonal arbors for doing the same thing. On the other hand, if you do have a retinotopic organization, then the axonal length you can see is much lesser. So one of the suggestions was that the map organization is an evolutionary solution, as it were, to the problem of optimizing the amount of wiring length that is required for achieving the same processing that would be required otherwise. Right? And this is an extension of this. It has been the same kind of argument has been used for multiple different maps where they would use different kinds of connectivity patterns. So this is the difference. So let's say there are two neurons sitting in the space over here, and the probability that they are going to be connected to each other is going to be governed by the difference in the orientation selectivity of these neurons that you are looking at, right? So it's not dependent upon distance. There is a distance component also, but the orientation difference is going to be predominantly determining as to what is going to happen, right? So if you constrain the wiring length with this kind of a connection distribution, then you would get a solution something like this. So the different colors represent different orientations, and you would get an organization which looks something like this. So experimentally what you find is something like this basically, where the authors claim that the overall structure in terms of the pin fields, in terms of the linear zones and so on and so forth have been replicated in the theoretical model. And what they do is they would go about doing it for different connection probabilities where you have a uniform distribution, you have a larger sigma and so on and so forth, and you will get different kinds of solutions. And the one that matches closest with both the actual experimentally measured orientation difference, the connection probability versus the orientation difference, and the map structure is something like this basically. I mean, so this matches and also this matches in terms of the overall qualitative features of the map that is sitting over there. So this is one of the hypotheses as to why you would need a map structure to basically reduce the connection, the wiring length, so you don't have to spend too much time in making these large connections that would be required if you have to perform a certain function at the specific location. That's one of the hypotheses. The other hypothesis is starts from Horace Barlow and even earlier. It is what's known as the efficient coding hypothesis. Right? So the postulate is that neurons form a natural, sparse, and distributed information theoretic coordinate system for natural images to efficiently encode the information that reaches them. So by efficiently encoding, it does not, they are very careful to distinguish between optimally encoding and efficiently encoding. So by efficient encoding, what they say is the neuron uses all the information that it is available to it in encoding whatever it has to do. Right? So it is as though it has a certain, say for instance, if you have the visual system and it is receiving inputs from the natural world, you have a certain statistics are associated with the natural information that is out there. Right? Say for instance, if you look at natural images, not something like this which is human made where you have like edges like that and so on and so forth. So if you have like a mountain or a sky or something or things like a natural scenery basically, then what you see is that the amount of low frequency component in those signals is much higher than the higher frequency components, right? So you have transitions coming very discreetly, and the slow transitions are much more prevalent than the fast transitions in such an image. So if you were to encode this statistics, if you want to incorporate this statistics into your encoding process, and you want to optimally encode this stimulus as opposed to some other thing which was made up of such such high frequency transitions all over, and you had say more jagged ones like this, let's say, where you have like large high frequency variations sitting over there. So then probably the visual system would have evolved differently. Right? So in this case, what is shown in this matrix of filters, this was the landmark paper by Bell and Sejnowski, where they presented these natural stimuli, natural visual stimuli, to an algorithm which would try to make the components independent, which is much more than decorrelation, and hence you are going beyond principal component analysis. And you are trying to find out which filters are required for you to be able to extract independent components of natural scenes. And surprise, surprise, it turns out that these filters look very similar to the orientation selective filters that sit in the primary visual cortex. Right? So if you look at this, so you have a certain, let's say these are like positive variations, and the black ones are negative variations, and the gray is 0. So you can see that it looks very similar to the orientation selective receptive fields. You can see various different orientation. This is 0, this is 90, this is 45, and so on and so forth. Some of them have more complex responses, which is also seen in the primary visual cortex. Right? So these filters looked very similar to the kind of filters that you actually see in the visual system for you to be able to get this. So you have the wiring rate wiring size optimization, and this to be coexisting to both efficiently encode and to reduce the amount of wiring that you require for achieving the same functionality. Right, so this is just an extension of the same thing. So there are, I mean, this is like a field on its own, efficient coding hypothesis and natural image statistics, and how neurons represent this natural image statistics is a field on its own. It doesn't restrict itself, of course, to the visual cortex. There are extensions to it to various different subsenses that are sitting over there. So it is as though this idea comes from Horace Barlow, as I said. So the neurons act as some kind of matched filter for the stimuli that they encode, and act as efficient coders of the incoming information, where you use the statistics of the incoming inputs for you to be able to design the filters the way you want it. It's like the filters are evolving over time in order to match with the statistics that is present out there for you to be able to do that. Right? So in other words, the optimality efficiency in neural coding depends on the statistics of what they decode. Visual neurons have different inputs from auditory system, and thus their coding strategies should reflect this inputs. So what we are saying is that if the auditory cortex were to receive visual inputs, they should start having the features that the visual neurons normally would have. So Murugan Kasur and his colleagues had done this experiment in 2000, where they rewired the auditory thalamus so that you get visual inputs rerouted to the auditory cortex A1 over here. Right? So normally your visual cortex V1, the primary visual cortex would look like this, where if you look at the response of neurons sitting on this cortical surface over here, you will see that there are nice pinwheels, different neuronal surface, neurons respond to different orientations. So these are the orientations, and you can see that there are pinwheels, there are linear zones, and so on and so forth. Normally you wouldn't have such structures sitting in the auditory cortex, but if you rewire and map the visual input onto the auditory neurons, which now get a new statistics of inputs that are coming over there, you start getting orientation selectivity over there. You also get pinwheels, you get linear zones, all the things that you see over here start appearing in the auditory cortex. So that is one way of experimentally showing this. The other way is to actively modulate the statistics of the information that is out there. Starts from the work of Hubel and Wiesel, of course. Right? So what they did was, they presented the animal with only one orientation during the critical period, and the representation for that single orientation is going to be higher compared to the other orientations because of this skewed statistics that you have in this rearing environment. This is an auditory equivalent of the same thing, right? So you have, if you look at the auditory structure and you map the different frequencies, you see that, the different frequencies which are shown over here, these are the frequencies to which neurons sitting in this area respond maximally to. And there is a topographic map over there, and if you specifically expose these animals to only 7 kilohertz, you see that the representation for that specific frequency has increased at the expense of frequencies in this range. So you can see that this is much smaller, and here the green is much larger. Right? So it's as though this is a much more sense-specific modulation paradigm, where you're trying to alter the statistics and you're trying to see if the neurons are matching this altered representation scheme when you do this kind of a experiment. Right? So you see that you do get some kind of a modulation in the statistics that you see over here. So this is what we saw from the sensory visual system and the auditory system sitting over here. So let's translate these ideas which have been postulated in the sensory map literature, the interneuronal map literature, to the intraneuronal map literature that we have been looking at. So if we translate to it, one postulate that you can formulate is that in this resonance frequency map that you see over here, if you translate this to the actual neuronal topograph that sits over here, it is as though the adjacent locations, the cell body and this part of the neuron, which is until around here, when the neuron rests around -65. So until around 200 microns, you see that this part of the neuron is almost responding to the same frequency. On the other hand, this distal end of it is responding to a slightly higher frequency. But if you look at this, the structure of connectivity onto this neuron, the stratum radiatum, as this cyan shaded part is called as, receives inputs from the CA3, and this purple, violet, whatever you want to call it, is the lacunosum moleculare, and that gets inputs from the entorhinal cortex. Right? Now consider a situation where we don't have any proof for this, but I will come to a certain indirect proof for that. Right? So consider a situation where the entorhinal inputs are at a slightly higher theta frequency range compared to the inputs that are coming from the CA3. Right? So, and you want a neuron which would respond optimally to a lower frequency for inputs that are coming from the CA3, and to a slightly higher frequency for inputs that are coming from the entorhinal. If your wiring was all messed up, where different parts of the entorhinal inputs also come over here and the CA3 inputs also go over there, you might not have been able to achieve this formulation where this whole part is having a 4 hertz resonance frequency. On the other hand, this part of it is having a 6 to 7 hertz resonance frequency, which is a large number, because if you look at the entorhinal cortex and go along the dorsoventral axis, there is a gradient of resonance frequencies, and that is basically this range, 4 to 6 hertz. Right? So if you were to have a different wiring organization, then you would not have been able to achieve this. But now, both because of the wiring diagram and because of the higher density of H channels sitting over here, you are able to achieve this specific selectivity to a specific frequency for a specific set of fibers. Right? So this is one way of looking at why these maps are formulated. The wiring diagram example, which is taken from the sensory map literature, and you could translate it back into this. So that's one way of looking at it. The second part, where we talked about plasticity in response to the statistics, the question that you first would ask is whether these intraneuronal maps are going to be plastic as well. The answer is yes. They can undergo selective plasticity only at the cell body, selective plasticity at one specific branch, and they can also undergo global plasticity, which spans across the somatodendritic gradient. So there are examples of the backpropagating action potentials changing at one specific location, the resonance frequency changing all over the entire somatoapical trunk, and so on and so forth. There are enough examples of things changing only at the soma and not other places also. Right? So you do have examples of plasticity occurring at the cell body. So the question is, what purpose do intraneuronal maps and their plasticity serve? Right? So, okay, go. So the hypothesis, which is again an extension from what we saw in the interneuronal maps, where the sensory regions were the system and the natural stimulus statistics were the input that is coming in, and you are trying to match the filters that are sitting over here to match with the statistics that is out over here in this faded yellow color thing, which is kind of invisible with this projector. Right? So on the other hand, if you look at the intraneuronal maps, where you're looking at different locations along the same neuron, you have the network statistics, the kind of, say for instance, the hippocampus receives theta frequency inputs. At different parts of it, you get different inputs coming from different locations. So the network statistics is what is going to be important. So the kind of network statistics that are present at that specific neuron that you are seeing at that specific location. So the single neuron is going to try to modulate its channel density in response to the kind of inputs that are coming at that specific location. It's not like a global thing. So because you can change ion channel densities locally at only one location, it is as though neurons are trying to encode this altered statistics that you see over here. If you formulate that, if you accept this hypothesis where the ion channels present in a neuron's compartments, their properties and densities are governed by the nature and statistics of the inputs that compartment receives, and are designed to efficiently encode this information. If you accept this hypothesis, the implications would be that what we see as learning, where you are trying to learn new things, and now it is accepted that you can also have changes in voltage gated ion channels as putative substrates for learning and memory. What we see as learning and as responses to pathological conditions are just consequences of this matching process. Right? So in the process of learning, there is a change in the input statistics to this neuron, and what this neuron is doing is trying to match with that, and that is permanently etched in that specific dendritic location, and hence learning could be posed as a mechanism by which it is efficiently encoding the inputs that are coming at that specific location. So I'm saying nothing new, I'm just translating things from the sensory literature to a single neuron standpoint, and reposing it from the standpoint of a single neuron rather than posing it from the point of view of a sensory region. Right? So the question then is, how are these maps formed in the first place? I mean, if it is important to match these statistics and so on and so forth, how is that the formulation or the development occurring? So in the sensory map literature, the modeling has been very successful in understanding and appreciating the impact of connectivity matrix within the cortical structure, let's say, over here. So within the cortical structure, adjacent neurons are connected by excitation, whereas slightly distal ones are connected by inhibition, leading to what is famously known as this Mexican hat structure. So this basically says that if there is a neuron sitting over here, the adjacent neuron is going to be probably connected by an excitatory input which has a weight something like this, and the neuron which is slightly away is probably connected by a feed-forward inhibition that is going to have a negative impact on this neuron. So if this neuron fires, this is going to shut down this. On the other hand, it's going to kind of enhance the probability of firing of this neuron. So this has been heavily used, and thanks to Cohen and others, the idea of self-organization where local computations and the interactions which are specific in nature between participating components, in this case neurons, lead to global order where the maps are formed in a certain order. So you get global order because not because you are enforcing it on the system, but because there are local computations and there are interactions between these adjacent compartments sitting over here, and hence you get global order over here. So here we understand this. We understand what kind of interactions exist between these different neurons and how they are going to translate themselves to structures like this where global order emerges as a consequence of local computation as well as the interactions between these neurons. So the obvious question to ask is, what kind of local computations and interactions drive intraneuronal maps? So this is with reference to the interneuronal maps that we have been doing forever and ever, and we understand some part of it, and we do know that there is some kind of Mexican hat connectivity sitting over there. So this has been able to explain at least to some extent as to what kind of impact does this connectivity and the local computations could have on this, and what impact does correlations between the neuronal activity and the environment have on this. These all these have been explored in detail, and if you look at, there is a review by Goodhill on in Neuron 2007. There was a special issue on maps. If you look at that, you will see that the impact of computation on the map literature has been really heavy, right? So on the other hand, from the standpoint of intraneuronal maps, we don't understand what are the local computations. We don't understand what the interactions are and how exactly they are contributing to the formation of this map that we see in these functional properties. So we came up with a simple process where we wanted to ask as to if there is a single cluster of ion channels present in this structure, which is a simple ball and stick Rawlish model, right? So you place a single cluster of voltage gated ion channels over here, and you want to understand that the rest of the entire structure is passive. You don't have any other ion channels sitting over there. So you place a single cluster of H conductance HCN channels at one location at 250 micron, which is halfway through this dendritic structure that you see over here. So it of course alters the input resistance at that specific location, because you are adding a conductance to that specific location. Of course it will change the input resistance in that location, and because it's a slow conductance, you will also see this typical Sag-ish response, which is the reason why you get that second order resonance dynamics. Otherwise it would be a simple Lorentzian sitting over there, right? So you have the input resistance at that specific location changing, all right, but it also alters the somatic input resistance, not by the same amount but by a smaller amount. It is as though the voltage gated ion channel sitting at one specific location along this topograph is capable of altering physiological properties not at that specific location but also at different locations at different points. So we quantified this, not the quantification, this is just the measurement. So you place, so that's the passive input resistance when there is nothing else, uniform RM, RA, nothing very fancy over there, and you will have an increase in input resistance with distance because of the cable property, because this is a sealed end at that end, and here you have the soma, which is a large compartment. So that's how the passive structure would look like. Now you are inserting H conductance of different densities there is only one cluster of that sitting at 250 micron. So you can see that it not only reduces the input resistance at that specific location, but it alters the input resistance at various different locations also. So if you now quantify the relative impact or the relative influence of this single structure at this location versus the others, then you will get something like this. Right? So you have this is the same plot that is shown over here, but normalized by the influence at the location at which you inserted that channel. Right? So now you see that irrespective of what the density of the channel was, the normalized influence factor, the normalized influence field is exactly the same across both sides, and you can quantify it by fitting exponentials on both sides, and you can arrive at the full width at half maximum, and that could be the spread of influence of this channel cluster across the dendritic topograph. Right? So if you do this, you will be able to understand as to what kind of interactions or ion channels sitting on the topograph are having on each other, and how they are going to be influencing others in arriving at the final input resistance gradient that you would see in a single neuronal structure, and what is the contribution of this influence. So if you just take, if you consider ion channels to be purely local, then they would have only influences at that location. On the other hand, if you have the spread added, what is the contribution of the spread? What is the contribution of that specific location alone? That is 40 or 45? 35, thanks. So you have, you can quantify that spread by measuring the full width at half maximum, and you can see as to how much the influence is. So you can see that even in a complex model, morphologically realistic model, if you place conductances of different densities, you see that it is going to be almost the same. The influence spread does not, so if you measure the full width at half maximum, it's not going to change much throughout. So you can see that the spread is not very different, because that is going to be dependent upon the cable properties and the other active channels that are sitting over here. Right? So you can also quantify as to what specific parameters would alter the spread in terms of how an ion channel sitting at one location is going to alter physiological processes, physiological measurements at different locations, and you can do a complete sensitivity analysis, which you can see in that paper that I showed you here. Right? So you can do that analysis, and you can also ask as to whether it is going to be measurement dependent. This is only input resistance, right? You can do it for resonance frequency, you can do it for backpropagating action potentials, which is here. So you can do it for backpropagating action potentials, where you place a single A-type potassium conductance at a given location and ask how much does it reduce the amplitude of action potentials. And here also you see that it is conductance independent, and you have the influence much smaller here compared to what you saw over here. And it will have a different sensitivity to the parameters associated with it. This is just showing the same thing, where you have an invariance to the conductance density that you see over here. Right? So the real question is not how much the influence is. The question is, how do these influences from different ion channel clusters interact with each other in coming up with that final functional map that you saw. Right? So in this case, where you have two different A-type potassium channel clusters, so one at 100, another at 350 sitting over here, when you have the separate influences measured, they look like that, the black traces, and you have the linear summation and the actual thing that you get by placing both these clusters together exactly match with each other. So in this case, for backpropagating action potentials, you see that the linearity index of this summation is almost 1. Right? So throughout, irrespective of where exactly your clusters are placed, the distance between these two clusters, you see that the linearity index is almost 1, suggesting that these influences summate linearly in arriving at the final map that you see. Right? So this is not true for all measurements. So if you have two 8-channel clusters placed over there, and you try to understand the influence fields for input resistance, that summation is going to be sublinear. So whereas two A-type potassium channels don't really talk to each other because of the dynamics of the kinetics of the action potential and so on and so forth, two 8-channel clusters do talk to each other and can influence the other in altering the physiological properties. It will not be linear as you see over here. So we took advantage of this linearity over here, and we asked the question that I just asked a few minutes ago, where you are trying to find out the influence of the spread as opposed to the influence of that specific, the ion channel sitting at that specific location alone. What would be the difference, which is more? So we just put together a simple convolution kind of a system, where we took the influence fields at different locations. So I have shown only few of them, but we took it at various different locations, right? and we had a gradient of A-type potassium channels, right? So this is the gradient in A-type potassium channel which we placed. So this is equivalent to having different clusters or different densities at different locations of it. Now what you do is you take the influence fields, multiply it by the effect of this A-type potassium channel on the BAP amplitude, do a convolution, and you will get a reduction which is something like this. So this is so this is the green one is the actual one, and the red one is the one that you get as an impact of this linear summation that you get, right? And the black trace is by considering only the impact of ion channels sitting at that specific location without accounting for the influences from other channels which are sitting at adjacent locations. So you can see that the amount of difference is pretty heavy. So if you take only the contributions of the ion channel sitting at that specific location, that doesn't give you what you get physiologically. This was by running the simulation as it is. This was from the linear reconstruction. So using the influence field. So you can see that that matches well, but if you consider only the ion channels sitting at that specific location, you don't get what you would expect from that, suggesting that the influence of ion channels sitting in adjacent compartments is cumulatively much higher than the impact of the ion channel sitting at that specific location, right? So we were able to do this only because of this linearity that is shown over here. Otherwise, we wouldn't have been able to do this, right? So here we have shown that you do have an influence you do have an interaction between adjacent compartments A, and you can reconstruct the map from this is the action potential amplitude with distance. It decreases with distance, and that is reconstructed by this influence field based linear reconstruction that you see over here, right? So we are able to show that there is an interaction A, and B, you can reconstruct the map from these interactions that are sitting over here, and C, the influence of adjacent compartments is really heavy and you cannot just ignore it, right? So it's as though you have local computations, all right, but the local computations alone will not be able to give you what you want. You need to have the interactions, which in this case are defined like this, where they are going down on both sides, where we have fit it with the exponentials in this case, but it is going down with the distance. And the combination of these two, these local computations along with these interactions, is leading to the map that you see over here, which is kind of very similar to what you saw earlier. But these are very early stages where you are just, okay, I'll come to that later. So this is the interneuronal map structure where you had adjacent neurons, and you had the Mexican hat interaction leading to cooperation and competition between different neurons, sculpting the interneuronal map sitting over there. So here what we have seen so far is basically only cooperation. You have to address competition by having, say, one regenerative conductance and one restorative conductance at different locations, and you will have to understand how that competition works in determining different structures. So this basically defines interactions between different compartments over here, and how an ion channel present at one location would alter the properties at other locations. That's a kind of simple quantification of what's happening over there. So what we are trying to do is we are trying to map the map structure that we have seen in sensory map literature, use the literature over there which is kind of rich and well developed, translate that mechanism to single neuron computation, and why these structures are present, why do neurons express so many different ion channels, why do they have to undergo plasticity. So these are some of the basic questions that we have which we don't have answers for. So these are basic postulates, and we are obviously a long way to go if we compare even with the sensory map literature, but we think of it as a first step basically, where you have tried to understand the thing, and so now we have to take it to the next step by defining competition B and try to understand if we can basically arrive at these map structures by placing constraints on the network architecture, the network statistics, and so on and so forth. So those are some of the questions that my lab addresses both theoretically as well as experimentally. So to summarize and conclude, so there do exist different functional maps within single neurons which are very analogous to sensory maps that span multiple neurons in the sensory system. Right? There are several functional maps that coexist on the same neuron. So we saw some examples, with each of them serving a different purpose. The idea is to find out how exactly they coexist, how exactly they interact with each other, and what computational power do they offer to this system as opposed to a simple passive neuron, which was the Kahalian viewpoint, right? So why do you need so many ion channels in the distal dendrites? I mean, why not have a simplified neuronal model where you have one sphere with multiple arrows, there is one nice nonlinearity sitting over there, just have an output sitting over there. So that's a very simple viewpoint, right? So you had passive summation, integrate and fire. Life is very simple. Why have all these nonlinearities sitting at the distal portion, changing the basic structure of the inputs that are coming inside to the neuron, to the cell body, thus altering the whole the structure of spatiotemporal integration in these neurons? Why have all this? So these are some ways of addressing these questions. Okay, where was it? So these interneuronal functional maps are mediated by channel gradients along the neuronal topograph. Different ones have different gradients. Different channels have different gradients, and each of them or multiple of them mediate the same functional map. Interneuronal maps are plastic, just as their interneuronal counterparts. There are enough examples in the literature now to suggest that ion channels can undergo localized plasticity in the dendritic location alone. You can have global plasticity throughout the entire neuron in response to certain activity patterns, where ion channels throughout the entire neuron will change either their the kinetic properties or their expression profile in order to match with certain activity patterns. So there is enough and more example over there. Putative physiological roles for these maps are wiring optimization and efficient coding, which basically we kind of quote unquote stole from the sensory map literature, trying to map if we could arrive at a solution of whether it is local wiring that it is optimizing, or is it efficient coding of the input statistics that is coming at that specific location that you see over there in that specific neuron, right? So different neurons obviously will have different kinds of maps, different kinds of ion channel densities, different kinds of ion channels expressed, and our suggestion is that that is going to be reflective of the statistics of the local network statistics at that specific input location, and hence it is different. So influence field to form a simplified quantification for the localized interaction between ion channels that result in the intraneuronal functional maps. Right? So acknowledgements, my mentor and collaborator Dan Johnston at UT Austin, Rahul in my lab who did most of the work, all of the work, and all the other lab members, and I have HFSP running my lab basically, without which I'll have to shut down. Thanks. Questions? Yeah.
I have a question on your influence fields, which I fully agree with the analysis and how they look. But it seems to me they would be much dependent on the local input resistance. So if you put in a big synaptic background, which would be increasing the local conductance,
it would basically
4 to 8-fold, they would shrink quite a bit.
It will shrink. So
shouldn't you look at those?
So what we have done is we have done a complete sensitivity analysis and asked questions about what would background synaptic, say for instance we would change RM, we would change RA, and relates that to say in vivo what would happen. You would have like a larger barrage of synaptic inputs coming onto these neurons, so thereby reducing the gain, the Abbott way, the neuron paper 2001. So you would have that kind of a reduction, and that would basically shrink the influence fields for input resistance. But for other things it would enhance it, like say for instance if you are measuring the influence fields of resonance frequency, because you are reducing the conductance, that would enhance the resonance frequency because you are shifting the filter characteristics such a way that you would have an enhancement in the resonance frequencies. So we have a complete sensitivity analysis which suggests the same thing that you have suggested basically.
I'm trying to understand the hypothesis you proposed, where you said that you want to reconcile somehow the input statistics with the ion channel statistics on the single neuron. I don't know what you are trying to optimize there. So that's what is missing.
So say for instance, let's assume that your distal inputs are getting theta frequency. I mean, you get theta frequency inputs at the distal end of a neuron, right? And they have, like, say, 7 hertz of, so it's a pure sinusoid, let's say, the theta frequency, if you measure the LFPs, they are pretty close to sinusoids actually. So when the animal is awake and behaving, you get theta plus gamma, theta riding on gamma. So those will look like nice sinusoids. So those are the kinds of inputs that are coming to the distal end of the neuron. Now what you want to optimize is the response of this neuron to that specific frequency and not any other thing, right? So what you do is, you keep on adding in your virtual world, you keep on adding additional 8 channels to that location, and we know that the resonance frequency, if you plot it on the y-axis and if you plot 8 channel density on the x-axis, that would look like a square root, going by the same kind of things that we know in electrical engineering and extending it to these things. So you will have a direct relationship. Okay, so you keep increasing this this density of 8 channels. That would make it optimally respond to that specific frequency. So at one optimal density of the 8 channels, you would have the response of that compartment matching optimally with the input statistics, right? So that is the relationship between the 8 channel density and resonance frequency in this case. But that's not the only feature that is that the neuron is encoding. There are different features, different ion channels are obviously encoding for different things. The question is, what is encoding what?
Okay, I think I still need to talk to you later about this, but then other thing is, let's say that I buy this hypothesis that you have an intraneuronal map, then why do I need a network? I can just, pass on everything feed forward, so I can basically think of everything going feed forward from sensory to the motor, and I don't.
So, but I mean, neurons unfortunately are not spherical cells, right? I mean, they have dendrites.
I'm not assuming. I'm buying your hypothesis that they are actually extended dendritic trees, and they have maps.
And you will also have to buy, I mean, you don't have to buy it, it is out there, that different parts of a neuron receive inputs from different regions.
Indeed.
Right?
I'm agreeing with you on that actually. So if it is like this, then you are bringing in some input, let's say from LG and P1 and so on. But then why do I need recurrent connectivity in the first place? If I have created all this effect of the maps within a neuron, why do I? So
you are looking at it from only the single neuron computation in this case, right? So
you are telling
so if you want to have feedback in terms of, say for instance, attention coming into the picture in modulating what you are concentrating in the LGN or in the V1, so which one you want to emphasize and so on and so forth, then you would need, so that is a network computation decision that you have to make, which is separate from what is going on in this single neuron. In this single neuron, you are worried about, okay, I am getting these inputs and I am trying to encode as efficiently as possible. To give you an example of what would be a sensory equivalent would be, say for instance, your visual system is encoding the natural scene statistics by having all those fancy orientation filters that are sitting over there, right? So now that doesn't account for your social interactions with somebody else, right? So that is a higher level thing.
I'm not talking about network at that level.
I'm
talking about layer 4.
So what I'm doing is I'm taking the system as one level and your social interactions at the next level. So the equivalence that I'm drawing is the neuron is at one level and the social interactions, quote unquote, that it is having at the other level. So we are talking about 2 different sets of scales of computation. So I'm talking about the scale at which we are looking at a single neuron, what kind of inputs that it is getting and how you are going to encode that, right? So not about how the network is going to be doing or what kind of network do you need for computing a certain thing. That is the kind of question that Chaklowski will ask. And he also doesn't ask basically that question. He would ask a question about, okay, if these are the computations that I have to perform, how efficiently can I make the wiring diagram so that I will be able to do the same thing with lesser amount of axonal length, right? So these are 2 different questions. We are talking, I mean, neuroscience research proceeds at multiple levels. We can't make a statement about the behavioral level by working at the genetic level, even though there are enough number of people who knock out one gene and put the animal into one behavioral task and do something. Yes, sir.
It's fun to see this perspective. But you're sort of, I'm intrigued that you're coming at it from the viewpoint of this would be optimal for the neuron to do. Whereas I'm curious to know, it seems to me that it would be highly constrained by what the molecular logic inside the neuron can do. Right? So there may be some kind of optimality that...
Yeah, I mean, so we are not necessarily saying that the features have to be as simplistic as resonance frequency or, I mean, say for instance in the visual system, if somebody had, if Hubel and Wiesel hadn't told us that there are orientation selective neurons, we wouldn't have probably recognized that there are neurons that are sensitive to orientation and there is a map of things sitting over there. So the question in the single neuron literature is that we don't even know in some senses as to what these specific locations are responding to and what kind of local computations they are doing. So that is something which we do not know. So there at least we had Hubel and Wiesel telling us, okay, there are orientation selective neurons, there are direction selective neurons, these are the features that you want to look for. If they hadn't told us that these are the features we want to look for, we wouldn't have possibly arrived at these maps in the first place. So
right, but you see, you cannot arrive at a set of features by saying these are desirable features.
Yeah, I mean, we don't know the features. I'm just giving you an example of resonance.
These are physiological feasible features. That is, the molecular logic permits such features to be created in the first place. So
yeah, so
it seems to me that that connection is still...
So the example that I provide in terms of the H channels and resonance frequency is just to make an intellectual connection of saying that, okay, this is a feature and this specific ion channel seems to be encoding for this, and there is a kind of relationship between this and that. That is the, I mean, that's just to make an intellectual connection. I'm not saying that all features have to be as simplistic as this. There could be more complex features. There could be second order relationships like what is being suggested now with that clustered plasticity stuff that we keep seeing. So there could be correlations which are being encoded. So there could be two different inputs coming into a specific dendritic branch, and there could be correlations that are encoded by the activation of, say, dendritic spikes at that location. So features could be more complicated. I'm not saying that this is the entire feature set.
Well, actually, I'm going the other way. I'm saying that even your feature set is probably too rich. That is, you cannot necessarily, let me rephrase that. You are still highly constrained by what the molecular mechanisms are available to establish these kinds of gradients of ion channels in the first place. Yeah? If you want a peak of potassium channels at a certain location, that has to be maintained by some molecular mechanism, anchoring proteins and so on. And I think that is going to be a huge constraint in even being able to devise such features.
Yeah, so, I mean, it is the usual top-down versus bottom-up conundrum, I would think. Right? I mean, a specific instance of that. What I'm trying to do is I'm trying to look at it from what feature, what broad thing that you want from a single neuron, and you find the mechanisms for doing that, or you try and find, okay, these are the kinds of mechanisms that would be required. There are instances where neurons don't have a gradient in A-type potassium channels, and there are specific instances where neurons do have a gradient which is maintained. And the example that I was going to state in support of the postulate that specific regions are going to be encoding specific inputs that are coming inside was one of the papers that came up 2007. It was a JBC paper where they lesioned the entorhinal cortex. So you have basically taken out the inputs that are coming onto the lacunosum, right? So now if this hypothesis were true, they obviously did not posit us from this hypothesis standpoint, then you would expect that the gradient that exists in the H channels, because those theta frequencies are gone now, and the gradient hence should be gone basically. And they indeed found that the Hessian-1 channel density has, you don't get the gradient anymore. So it is driven by the activity patterns that are coming from the entorhinal. So it's an indirect proof, but I mean it supports the hypothesis. Of course, in science we don't prove anything, right? I mean we either disprove something or we add evidences in favor of some hypothesis. So that is evidence in favor of the hypothesis that the channel density at the distal part of the CA1 pyramids are driven by the entorhinal cortical inputs that come onto the distal locations. So that was one of the, basic things I wanted to state, but I forgot it. Thanks. So it's basically the molecular mechanisms are driven towards making a certain kind of computation that the neuron is supposed to do. That is the broad way of putting it. Again, from the top-down way of looking at it as opposed to a bottom-up way of looking at the same thing.